everyone. Can you see me? Can you give a comment? Welcome to BiochemSerye. This is our first live stream. And can I have a shout out from where you are watching? Where you're watching from? I'm from Quezon City. Can you comment on the chat box? So I think we have 6 viewers now. So thank you very much for coming. By the way, I'm Dr. Noel Martinez Bautista, and everybody calls me Doc Bao. No, I've been teaching biochem for 25 years. Oh my God, 25 years. But I just recently retired. Okay, but teaching is in my blood, so I'll try to maybe share this to more people via the powers of YouTube. So thank you very much. No, this is historic because you're my first guests. Anyway, what are we going to discuss tonight? So welcome everyone. Welcome to Biochem Seria. This is our first live stream. Wait, I'll share my. Okay, so this is our first live stream. I'll make it bigger. Okay, so wait, okay, there. Okay, so what we're going to discuss for tonight is a topic that confounds a lot of students. Okay, for the decades and years that I've been teaching, students really have a problem in memorizing, actually in remembering the amino acids, because here you have a lot, right? We'll be discussing later on how many they are. Okay, I've been searching the web far and wide, and I've seen a lot of wait. Okay, I've seen a lot of mnemonics. Okay, so confusing mnemonics. Mnemonics supposed to make you remember a lot easier, right? But remembering the mnemonics sometimes will make it harder because you don't even know what the mnemonic stands for, and mnemonics supposed to be helping aids. Okay, but the best way really to study chemistry of amino acid is to look into the structure. I know there are a lot of them. No? You have more than 10, you have more than 18, you have more than 19. But if we're going to look at the structure and remember them, no, there's always an easier way. It will be easier, and you don't have to perplex yourself remembering all these mnemonics. Okay, so can Jacobs, where are you watching from? Okay, so Jacobs, hello po, Doc Wewe, yes. A lot of people call me that. Okay, so where you watching from, Jacobs? Anyway, this will be interactive. Okay, so I'll be posting questions later on. Don't worry, they'll not be graded. Okay, and you can ask questions. Okay, if there are things that needs clarifying, no, and I'm sure there are a lot of them, feel free. Okay, feel free to. Hello, Kim. Okay, good luck on your medical board exams. Okay, so feel free to post. Okay, hopefully, I'll do my best, Kim, to make it high yield. Okay, so Jacobs, no, from Coronadal. Okay, South Cotabato. That's far. Okay, so we have Andrew Reyes from Laguna. Thank you very much, no, for dropping by. Anyway, so let me proceed. So this is what we're going to discuss tonight. Okay, I know there are a lot, no, but I'll try to keep this under under an hour. Okay, so very briefly, I'll look into the different functions, okay, of your amino acid chemistry. Amino acids, no, there are a lot. Some say that they're the most versatile. Okay, they're the most, not really the most diverse, no, but they're the most versatile. Then this is what is really very important. Okay, we will now look into the chemistries of your amino acid. No, they have a common formula, but we will look at what will confer to them specificity or uniqueness, no, in their chemical structure, in their properties, and in their reactions. Okay, and since you have a lot of amino acids, and you know very well that amino acids, they make up your polypeptide chains, no, that can be very long. So you might have hundreds, even thousands. So it might be too tedious if you put in the whole name of the amino acid. So our forefathers in biochem, no, they made it easier. Okay, but you have a little more things to memorize, but a lot more easier for the whole of society. Okay, then we're going to classify. Okay, the confusion really in amino acids, and you can see this in questions. No, in questions, is how they are classified. Okay, if you will look into this now, you have different mnemonics, okay, for the classification of your amino acids, okay? Because say your amino acids are classified in too many ways. No? They're classified into their polarity, they're classified into their structural features. We will look into that. Then you know very well that amino acids can be essential or non-essential. Okay, so we will classify them. And ito, no, so that to put value into what we have learned, hopefully no, we learned something. And did you get something out of this lecture? We'll have practice exercises. So these are actual questions no, from a certain medical school in Manila several years back. Okay, so by the way, if you find value, hopefully if you find value in this lecture, kindly click on the like button, then subscribe as well, and click on the notification bell. Okay, so let's move on. For the functions of amino acids, we'll make this fast. Don't be intimidated. Okay, I know you're seeing a lot. Okay, you're seeing a lot. Okay, sorry, Andrew. Yan. Okay, so we'll hide you. Okay, so from here you can see a lot, and it might be confounding. It might be perplexing. No, but let's just concentrate ourselves here because from here you can see a summary of the functions of amino acids. Amino acids, they have a myriad. They have a lot of functions. Okay, they have a lot of functions. But primarily, if you're asked in the boards, or if you're asked in the exams, or in MLE, etc., if you're asked what is the primary function of amino acids, anyone, what's the primary function of amino acids? By the way, amino acids, the primary function is they're proteinogenic. No, they are absorbed and they are taken into the body to produce protein. So that is their primary functions. But it does not end there. Okay, your amino acids, they serve a lot of function. No, they are the building blocks for a lot of things. From here, you can see that GABA, that epinephrine or epinephrine, they're the building blocks of your hormones. Okay, then they're the building blocks of your nucleotides as well, no? Your pyrimidines and your purines, your thyroid hormones, your melatonin, serotonin, no? Your coenzymes. So proteins, they serve a lot of function as well. But later on, no, when you're going to discuss amino acid metabolism, you'll find out that amino acids, no, in their situations, for example in severe starvation, amino acids are not only the building blocks, no, of proteins, but they can also be what? Source of energy. Okay, they can be a source of energy. Sometimes, or maybe in the past, you might have remembered or you might have heard of glucogenic and ketogenic amino acids. No? So glucogenic, no, they will now form your carbon skeletons that can enter, okay, your Krebs cycle, your TCA, okay, to provide additional energy, no, for dire situations. Or they can form, no, your ketone bodies. They can be emergency sources of energy as well. Okay. Then your amino acids are also what? No, they're the transport forms of your nitrogen. Okay. So with that, we can now proceed to your amino acid chemistry. Okay. So this is really very basic. Okay. So amino acid chemistry. Why are they termed amino acid? Because they're the amino. Okay, the amino group of your carboxylic acid. Okay, so sometimes they are called your 2-amino carboxylate, not 2-amino carboxylate or carboxylic acid. Okay, so this is your alpha carbon. Okay, this is your alpha carbon that is the central atom in your amino acid. Okay, and just like your hand, no, this is chiral. No, chiral comes from the Greek word kairos, meaning hand, because you have different fingers, right? You don't have two thumbs, etc. So just the same way, no, your alpha carbon in your amino acid, they are connected to four different substituents. Okay, so all of your amino acids except for one will have four different substituents. Okay, so all amino acids will have your carboxylate group, and we'll look at this later on. Your carboxylate or carboxylic acid, sometimes not they are written as COOH, okay, in their unionized form. Then you have your amino group, okay, amino group, or your NH3 positive, the ionized, or sometimes it's just NH2. Now we'll look at that later on. And your hydrogen, okay. So all your amino acids will have these three, okay. Your hydrogen, okay, your carboxylate or your carboxylic acid group, and your amino group. and they are attached to your alpha carbon. Is alpha carbon your carbon 1? Remember this. You remember this? Your carbon, your alpha carbon is carbon 2. No, it is always your carboxylate carbon that is carbon 1. So later on when we're going to count, no, when we're going to count the positions of your substituents, we should always remember that, okay? The alpha carbon is the carbon that is attached to your carboxyl carbon, okay? Attached to your carboxyl carbon. So that's that. And you have something that gives uniqueness, no, to your amino acids, and this is your R, or this is your side chain. Okay, so that's your R. So your R group, or sometimes known as your excuse me, your side chain, they will confer now your uniqueness, okay, your uniqueness in the structure of your amino acids, and consequently it will confer uniqueness in their property and in their bonding, no, or in their reaction intramolecularly or intermolecularly, no, with other molecules. Okay, so since we're dealing with acids, no, from the term acids, no, amino acids, I think we cannot escape studying this concept, no, your Bronsted-Lowry concept of acids and bases. Okay, so you know your Lewis, no, you know your Bronsted-Lowry. Kaya sometimes, no, your they're simply called as Bronsted acids. Okay, so what is a Bronsted acid? A Bronsted acid is basically what? Now, what is being exchanged or what is being given away? Anyone? What is being given away by a Bronsted acid? A Bronsted acid will just give away what? A hydrogen ion. Okay, a hydrogen ion. And a Bronsted base, on the other hand, is an acceptor of your hydrogen ion. Okay, so what is a hydrogen ion? We know very well that a hydrogen atom is what? You have a proton and an electron, right? That is the simplest of your, wait, I show myself. Okay, that's the simplest of your atoms, right? That's atomic number one. However, if it is a hydrogen ion, basically what is that? A hydrogen ion is just a proton. Okay, so a bronzed acid is something that will give away or that can donate a proton. That is a bronzed acid. Okay, because later on I'll be maybe I might go into live stream again, no? Because in BiochemSerye, I tried recording. Okay, I tried recording lectures, but for me it seemed natural, and not I'm not really that comfortable. So maybe my next live stream will be on the ionic properties of amino acids, no? Which is another difficult topic, no, for a lot of students. Okay, so a bronzed acid is a moiety that gives off that can donate a hydrogen ion. On the other hand, a Bronsted base is something that can accept. Okay, something that can accept. So if we're going to look at the amino acid, which of the following is the Bronsted acid? No, the Bronsted acid here is what? No, your Bronsted acid is your carboxyl group, right? So if you're going to look at this, okay, your COH, okay, that's your carboxylic acid. However, if it gives away its hydrogen ion, which is a positive, no? It's a cation, it's a proton, it becomes your carboxylate. Okay, so please take note of your semantics. Okay, if it is carboxylic acid, this is usually the still you still have your hydrogen. But if it is your carboxylate, it's in the ionic form. Okay, so that is your Bronsted acid. So for your amino acid, the Bronsted acid is your carboxyl group. Okay, so if it donates hydrogen ion, which is a proton, which is a cation, it becomes negatively charged. So if you're asked a question, now which of the following amino acids will have a negative charge? No, which will have a negative charge once it is ionized? No, so I always think of an acid, okay, because it gives off a hydrogen ion. On the other hand, in the same vein, what is a Bronsted base? No, that's why your amino group is also known as your basic group or your Bronsted base, because it can accept a hydrogen ion, then it becomes your ammonium, no, your ammonium moiety, your NH3. Okay, so that's that. So if we're going to look at this, no, so this is a diprotic amino acid. In other words, it has only two functional groups. Okay, I forgot to mention that all amino acids have at least two functional groups. Okay, in this case, that's your carboxyl group and your amino group. So this is what is this. Now we can see that this has donated the hydrogen ion. So your carboxylate is the Bronsted acid. Once it donates a hydrogen ion, no, a proton, it becomes your carboxylate. Okay, a carboxylate. On the other hand, your amino group, this is your Bronsted base. Once it accepts a proton or hydrogen ion, it becomes your ammonium. It's positive. Okay, so in this case, no, this is your cation. This is your cationic species. This is your anionic species. In the middle, no, in most amino acids, no, disregarding the R group at physiologic pH, not 7.4, all amino acids will exist in a dipolar, okay, in the doubly charged, okay, in a doubly charged moiety, but with a charge of 0. Okay, so it has positive 1 and it's negative 1, but the overall charge is 0, and you call this your sweeter ion. Okay, so you have your cationic species, you have your anionic species, and you have your sweeter ionic species. Okay, we'll be discussing this in a later live stream, no, the ionic properties, okay, which is really more complicated, but this is just an introduction. Just finished discussing proteins in a biochem class. Okay, so thank you. Okay, so John Beltran. Yeah, and I hope this will help you. Okay, remember, no, because amino acids really that complicated. So thank you very much, John Beltran, for dropping by. Okay, so just like your carbohydrates, so you just finished with your carbohydrates. Okay, since your alpha carbon is a chiral carbon, no, it's like a hand, no, four different substituents, you can have enantiomeric forms. Okay, so in Fischer projection, no, Fischer projection is not only for carbohydrates, it is for amino acids as well. Okay, for amino acids as well. What is the for carbohydrates, the carbohydrates exist in your L and your D enantiomeric forms, right? In your amino acids, the same. Okay, they're in enantiomers. Okay, they're enantiomers. You have your L amino acid and you have your D amino acid. For carbohydrates, if anyone can remember, what is our benchmark, or what is our miles? What is our reference? What is our reference? How do we know if a monosaccharide is a D or an L enantiomer? Usually, we look at the hydroxyl group that is attached to your penultimate chiral carbon, right? Right, the second to the last. Okay, but in amino acids, no, basically just similar. But make sure, please remember that the most oxidized group, your carboxylate group, should be pointing upwards. Okay, should be pointing upwards. So once it is pointing upwards, what do we now look? Is we look into the location or the orientation of the amino group. Okay, the orientation of your amino group. If the amino group is oriented not to the left, it gives us a it makes it's easy to remember. If it's oriented to the left of your chiral carbon, this is your L amino acid. Okay, so consequently, if the amino group is oriented to the right of the carbon, right, that is your D. Okay, if you can see, it's inverted, right? It's flipped. So it's really, no, it's really theoretically a mirror image. Okay, a mirror image. But if you rotate this, if you rotate the L amino and try to superimpose it to the D amino, it will not. It will not. It will not jive. It will not superimpose. They're non-superimposable, just like in your carbohydrates. Okay, so basically that's that. Which do you think ito? One of two choices. Amino acids are present in which enantiomeric forms? In nature. Basically, predominantly in which enantiomeric form? L or D? Anyone? L or D? Okay, so it's the opposite of your carbohydrates. Okay, carbohydrates is what? The carbohydrates is D. Okay, but amino acids, it is your L. Okay, so most, no, predominantly, almost all in nature. Very good. Okay. Yes. No, Joel, no, you're right. You have a question mark, but you're right. Okay. But you're right. Just think of life. Okay. Proteins is life. Okay. Proteins is life. Gives you muscles, etc. Okay. Protein is life. Just don't 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 mix the two. Okay, so amino acids is left L, and carbohydrates is D. So the basis here is the same, no? Carbohydrates, monosaccharides. The basis is also glyceraldehyde. Okay, glyceraldehyde. Usually the horizontal is from the page pointing to us. Okay, pointing to us. The vertical lines are what, away from us. Okay, so that's just a review. Standard amino acids. If I'm going to ask you a question, how many standard amino acids do we have? By standard, meaning they are translated by the genetic code. You have your codon, okay? You have your triplet. You have a codon that will code for this, okay? So they are translated. How many? I know we have hundreds of amino acids, but we have only how many standard amino acids? Anyone? Anyone? You're not graded, okay? So feel free. And I'll just shout out. I'll just show who will. Um, John, I know where you're coming from, okay? I know where you're coming from because I know you have your 21st and you have your 22nd, no? But theoretically, I'll show this later on. Huh? Theoretically, strictly speaking, okay, you have what? You have same number of hands, fingers, and same number of toes, no? Dok, medyo kadiri, no toes, no, but we only have one set, no, we only have one set of hands, and so just remember how many toes and how many fingers you have. Very good, no, Alvin, you're right, no, so you have 20. So I'll show this later on. I know some of you will, Doc. I know there's a 21st and there's 22nd. I know we will discuss that. But from the latest, okay, from the latest, it's okay. It's okay. It's okay. It's okay, John. But you're partially right. You're partly right. Huh? You're partly right. Okay. So Alvin, you're right. Okay. So you have 20. So that you'll remember until the day of your MLE exams, until the day of your endocrine subspecialty board exams. As far as I know, as far as I know, I've looked this up, you still have 20. Okay. Okay, so I know, no. So so you can assign, no. Okay, you can assign one set. Okay, you can assign one set of amino acids per group of fingers. Okay, so we'll go to this one by one later on. Okay, so first is you have five. Okay, we'll show what is common to all five. No, these are your aliphatic. Okay, these are your aliphatic non-polar amino acids. Okay, then you have what if we have aliphatic, meaning by aliphatic, no, to remind everyone, aliphatic means you don't have any cyclic structures, no, you don't have any aromatic rings. Okay, and later on we will see that the R groups are not heteroatoms, so they don't have any oxygen, they don't have any sulfur, they don't have any nitrogen. It is just carbon and hydrogen. So that is the first group. Okay, so that's your aliphatic. Then the next is aromatic. No, aromatic. We look at the structure so that you can remember it more easily. Aromatic are the cyclic structure. No, most of them they have the benzene ring or the phenyl ring. Okay. So the next group is, we'll see this later on. Okay, these are serine and threonine. Doc, what's common among the two? No, we'll see this later on. No, so this is they have a similar functional group. They have a similar functional group. Then cysteine and methionine, I'm sure what is common with cysteine and methionine? What's common with cysteine and methionine? Anyone? They are what? No, they contain what? They have a sulfur atom. Okay, they have a sulfur atom, and you know very well, no, aspartic acid and glutamic acid. Okay, Doc, these are three-letter names. We'll go to the nomenclature later on. No, these are your acidic amino acids, and you have your amides. They're amides. No, your asparagine and glutamine. If you have acidic amino acids, you have your basic amino acids. No, this might be controversial. No, but most books will give these three. Histidine, arginine, and lysine, okay, are your basic amino acids, and you know very well what basic means. They have a nitrogen that can accept a hydrogen ion in their R group. Pag acidic, they have an additional carboxyl group that can donate a proton. And what's the last? Can anyone give what's the bunso? What is the bunso? Very, very special amino acids because it is not an alpha amino acid, okay? It is an amino acid. It is an amino acid. It is your proline. Okay, it is your proline. So let's look at the nomenclature. Ah, yeah, no. So before I go, no, somebody answered, no, yan, si John Ancheta and si John Lester. Okay, 21 and 22. No, this is the latest. Okay. Um, Again, no, to define a standard amino acid is an amino acid that is what translated. Okay, that is translated from and has a genetic code. Okay, and you don't need any post translational modification. Maybe, I'm sure you have heard of your hydroxylation and hydroxyproline, right? Hydroxylation and hydroxyproline, they are in collagen, for example. They are modified after being translated, no? After being synthesized, no, in the ribosomes. However, your selenocysteine, no, your selenocysteine, no, you have your it's like cysteine, okay, but instead of sulfur, no, you have your selenium. Okay, and your amino acid pyrrolysine. No, this looks like lysine. We'll look at lysine later on, but it is a pyrrole ring. Okay, that's of 21st and 22nd. No, very rarely they will be translated. They will be translated by the stop codons. Okay, by the stop codons. Your uwag, uga, uwa. Okay, your UAG, UAA, etc. No, and that's that. Okay, if somebody, but as far as I know, no, you can correct me, no, if I'm mistaken, the number of standard amino acids are still 20. Okay, so how do we name them? You have three ways. No, you have three ways. You name them by their full name. Okay, Doc Noel Martin Bautista. Okay, sometimes I call Dr. Bao, no, BAU. So you have your three-letter name, and because you might have polypeptides that have thousands, okay, thousands of amino acids, it might be very tedious, no, to string them together with their full names, okay, and even with their three-letter names, so they devised a one-letter name. Okay, so very simply, okay, except for some, no, except for some exceptions, except for some amino acids, the three-letter name will just correspond to the first three letters. Okay, will just correspond for the three for the first three letters. Okay, so for alanine, cysteine, aspartic acid, glutamic acid, phenylalanine, this is pH. Okay, this is to remind us later on. Glycine, histidine, isoleucine is different. No? So you have that exception. Lysine, leucine, methionine, asparagine is different because you have your aspartic acid. Okay, so just take note. Okay, just take note. Proline, glutamine is because glutamic acid is GLU. So one book I remembered is that the first two letters is constant. Okay, the first two letters will stand for the first two letters of their name. And for the more common amino acids, the third letter will stand for the third letter of their full name. Okay, then the third letter for the exceptions usually is it sounds like what is emphasized. For example, asparagine, so that is ASN. Okay, glutamine, so that's GLN. Okay, arginine is the same, ARG. Okay, serine, threonine, no problem. Valine, no problem. Tryptophan, because this is confusing, no? Tryptophan, they did not use TRY, okay, because you have your TYR for tyrosine. It might be confusing, okay, if you have tons and tons of amino acids. So here is tryptophan, okay, so what is being emphasized? So please take note, okay, don't misinterpret or interchange the third letter. Okay, and this is what is confusing, no? In polypeptide chemistry, no, they use the one-letter names. Okay, so for the most common, they use the first letters. Okay, so G for glycine, H for histidine, T for threonine, S for serine, P for proline, no problem. But you have a lot of amino acids. You have amino acids that share the first three letters, right? You have how many amino acids that start with the letter T? You have, I think, three, right? You have three. You have threonine, tyrosine, tryptophan, right? How many starts with the letter A? You have alanine, you have aspartic acid, you have asparagine. How many starts with the letter G? You have two. You have glutamic acid, you have glutamine. So they have to assign other letters. Okay. So in this case, just for you not to remember, no, you just pronounce them as aspartic. Okay, aspartic acid, glutamic acid. Okay, glutamic acid. We Filipinos, no, we, we have problems with this. Phenylalanine, okay. F, okay. So that F, this is a little stretched, okay. It's a little stretched. Letter K, no, you don't have any letter K in lysine, but they say this is the letter that is nearest to the letter K, okay. Sorry, letter K is the nearest to the letter L, okay. So Doc, that's really stretching it too far. Okay, I know, but you just take it as it is. Okay, so hopefully, if you remember this, now you emphasize this K, A B C D F G H I J K L. Right? So K is for lysine. Okay. Asparagine again. No, remember no what is being emphasized loudly. Okay, asparagine. Then glutamine is cutamine, cutamine. Okay, arginine. No, so arginine. So that's letter R. And this is one tip, okay? Tyrosine, why? No, maybe I don't know. No, just remember why is for tyrosine. Tryptophan, an additional. Doc, why did they choose W? Does it sound like tryptophan? No, but they say that tryptophan is the only amino acid that has a double ring. Later on we will see. Okay, that has a double ring. So double ring. So if in the exam, if you're given which of the following is a double is tryptophan, and you have all the aromatic amino acids, oh my God. Okay, just look for the amino acid that has a double ring. Or you see you have all the aromatic amino acids, then you look for the actually, the double ring structure is your indole. Okay, it has a six-membered and a five-membered. No, that is your indole. It is a double ring. Okay, so I hope that helps you remember. Okay, is this helpful? And when is this helpful? Okay, so that's that. Huh? So if we move on, okay, let's now go into the meat of the matter. Okay, so first is your first hand. Okay, you have five. You have five. Again, what do we mean by aliphatic? No, aliphatic means you don't have any cyclic structure. You don't have any cyclic structure. So, by the way, what's special with glycine? 19 of the 20 amino acids have what? Have four different substituents, right? But in glycine, what is the R group in glycine? The R group in glycine is age. So what is conferred to glycine? What can we say about glycine? What can we say about glycine? Glycine has no chiral carbon. Okay, glycine has no chiral carbon. So it has no L or D enantiomeric forms. Okay, so glycine has no L or D. So if it's glycine, it will just be mentioned as glycine. Okay, and what did we say again about aliphatic? Why are they non-polar? They are non-polar because you don't have any heteroatoms, no? You don't have any oxygen, you don't have any nitrogen, you don't have any sulfur. Okay, so you have only carbon and hydrogen, carbon and hydrogen. And I had a video that I uploaded, no, on the chemical reactions, on the introduction to biochemistry. Okay, what can we say about the electronegativity between carbon and hydrogen? Electronegativity between carbon and hydrogen is what? They're almost the same. So electronegativity is the degree of attraction for the electrons. Okay, the degree of attraction for the electrons. So in this case, the electrons are shared. They are nonpolar. No, they're nonpolar. The hydrogen here cannot participate in hydrogen bonding. Okay, so aliphatic, they are all nonpolar because they are only the R groups are composed only of your carbon and hydrogen. If it's only composed of carbon and hydrogen, the electrons are shared equally. Okay, so that's that. No, so nonpolar. What can we say? If there's a question, um, the aliphatic nonpolar amino acids in a globular protein, okay, in a globular protein will locate themselves inside on the inner or exposed to water. So they're aliphatic, they're nonpolar, they are hydrophobic. Okay, they're hydrophobic, so they will be found inside, not within. Okay, they will be found within the molecule. Okay, so glycine, alanine. Okay, so just proline we'll discuss later on proline. Okay, so this five first. Now this five. Okay, valine and leucine. What is special among the three is what they have branching. Okay, they have branching. They are branching. Okay, they are branching. You learn in biochemistry if a structure is branched, they are harder to synthesize. Okay, they're harder to synthesize. So that will give you a clue. This is a branched chain amino acid. This is harder to synthesize. So they are what? Later on, we'll go to essentiality. All branched chain amino acids are essential. Essential, they are needed in the diet. Otherwise, you'll be deficient because your body cannot synthesize such. Okay, so that's already five. Okay, that's already five. Glycine, alanine, valine, leucine, isoleucine. They're non-polar aliphatic. I think this is a lot better than memorizing the mnemonics, no? Because it's just ultra short-term memory. After the exam, you forget them. Then the next exam, you'll remember them again, and it's not really, for me, that's not the way. Okay. Aromatic basically is what? No, aromatic. You have a benzene ring. Okay, you have a benzene ring. You have a benzene ring. From organic chemistry, you know very well that your electrons here are delocalized. No? They are shared equally. Okay, they're shared equally among the carbon. So sometimes you don't put the lines anymore. You just put a circle. Okay, so since the electrons are shared equally, you know, this is really hydrophobic. Okay, phenylalanine is one of the most hydrophobic amino acids. Okay, so phenylalanine is nonpolar. Okay, phenylalanine is nonpolar because um, your aromatic rings will only have some degree of polarity based on their substituents. As you can see here, substituents. Now, if the substituents have nitrogen or oxygen, no, like tyrosine, okay, then they will become polar. Tyrosine is what? It's just a hydroxylated phenylalanine. No, in the body we can easily hydroxylate your phenylalanine, so this is your tyrosine. So tyrosine is essential. Okay, phenylalanine is essential. Tyrosine is non-essential. Okay, I'm correcting myself. This is non-essential. You can easily hydroxylate your phenylalanine to tyrosine. Okay, so take note of the one-letter names. Okay, in books, no, if you use the three letters, no, they are all in small letters. Okay, they are all in small letters, but the one-letter name is in capital. Okay, tryptophan. What is the one-letter name for tryptophan? Is W. No, as I mentioned, it is the only amino acid that has a double ring. Okay, that's a good tip. Okay, so it has a double ring, and it is the indole. And this is the indole. Okay, you're not seeing any substituents, okay, that can ionize, etc. Tryptophan is non-polar as well. Okay, it's non-polar as well. Tryptophan, this is a hard amino acid to synthesize. Anyway, we have a mnemonic. No, the only mnemonic that I'm going to use. Okay, the only mnemonic that I'm going to use now for your essential amino acids. So for the asterisk, please take note. If you have a ball pen, if you have a pen and paper, please do remember. I'll ask you to remember 7 amino acids that really have to be remembered. Okay, tyrosine. What is the functional group that can ionize? No, that can ionize. This is your phenyl hydroxyl. Okay, so in this case, you don't only have two functional groups. Now you have your carboxyl group, you have the amino group, but you have a third functional group. Okay, so this is a trifunctional amino acids. This is a trifunctional amino acid. So later on, when we go to the polarity, now when we classify them, if they're neutral, acid, or base, now take note, tyrosine is acidic. Okay, slightly acidic. Serine and threonine, what is common? What is common? Serine and threonine, what is common? These are what? No, your serine and threonine. So this is we're almost, okay, we're almost 10, no? We're almost halfway. Serine and threonine are what? They are hydroxyl containing. Okay, right. Okay, they are the hydroxyl containing amino acid. They're hydroxyl containing amino acids. So as such, they are hydroxyl. They don't have any aromatic rings, so they're aliphatic, but they are polar. But they are uncharged. Which moieties will give a charged group? No, once they ionize, these are your acids and bases. Okay, these are your acids and bases. These are your acids and bases. Okay, acidic amino acids and basic amino acids. So so by the way, no, so these are your serine and your threonine, so they are polar, but they are uncharged. In other way, it means that they are what? They are non. Sorry. They are neutral. Huh? They are neutral. They are neutral, but they are polar. Because later on we will see that amino acids are grouped into acidic, basic, neutral. But neutral does not mean they're always nonpolar. Neutral can also be polar and nonpolar. Okay, so later on we'll group them as such. Then your sulfur containing, no, it's gonna go to the fit. Okay, so 11 and 12, cysteine and methionine, they contain sulfur. Okay, they contain sulfur. By the way, before I forget, okay, threonine, what do we see in threonine? It's a branch. Okay, it's branch chain. Branch chain. It's branch chain. So sometimes, no, you might be asked which of the following is a branch chain, and you don't see valine, leucine, and isoleucine, but you see threonine. Some books will classify threonine as branch chain. Okay, yeah, it's branch chain. No? You have a branch chain. Okay, so that's an additional thing to remember. Sulfur containing your cysteine, you have a sulfhydryl group. Okay, sulfhydryl group. You have an asterisk. Okay, so please do remember this. This is another trifunctional amino acid. A trifunctional amino acid. It has a third functional group. And what do you call this? This is your sulfhydryl group. Okay, that is your sulfhydryl group. But in this case, sulfur we know very well is a highly electronegative. Okay, it's a highly electronegative atom. Sulfur is a highly electronegative atom. So the electrons are mostly what grabbed by sulfur. So hydrogen can go away, can be donated. So cysteine is what is classified as acidic. Okay, cysteine. So if you remember the structure, I know it will take some effort. Okay, um, just take the effort. Okay, you look into the structure and you'll remember more easily. Okay. By the way, if you're getting some value, no, you might consider subscribing, okay, or liking. No, please like this video as well. Methionine, methionine is what? No, sulfur is electronegative, no, but it has already been shielded by the methyl, no. I think this is your thiol S ether, no, thiol ether. So your methionine is non-polar. Okay, so you have only two sulfur-containing amino acids, but you can see that they're worlds apart. Okay, this is what? No, this is methionine is non-polar. No, it's bulky, it's hydrophobic, it's non-polar, but it is what? It is essential. No, this is hard to synthesize, impossible to synthesize. However, cysteine is weakly acidic. No, weakly acidic. It means that it is polar, but it can be synthesized from the body. Okay, actually, when you synthesize cysteine from glycine, no, you need the sulfur that will be donated by or that will be sourced from methionine. Okay, so now acidic is what do we mean by acidic? You have a additional carboxyl group. You have an additional carboxyl group that can donate a hydrogen. Okay, please do remember, okay, you only have a methylene group. That is the difference. No? So, by the way, I forgot to tell you about counting. No? If this is your alpha carbon, okay, if this is alpha carbon, what do you call this carbon? If we're going to count by using numbers, okay, this is carbon 1. The carboxylate carbon is always carbon 1. Okay, please do remember that the carboxylate carbon is always carbon 1. This is carbon 1, this is carbon 2, this is carbon 3. Okay, but sometimes no, when you give the scientific name, no, if you're asked what is the third functional group of your aspartic acid, so this is alpha, this is your beta. This is your beta carboxyl. Okay, that is your beta carboxyl or 1, 2, 3 or 3 carboxyl. Okay. On the other hand, glutamic acid or glutamate, no, glutamate, this is aspartic. Okay, aspartic acid. Okay, this is glutamate. Okay, glutamate, that is letter E. What is the third functional group? This is alpha carbon, beta carbon. Okay, alpha, beta, gamma. Okay, so this is your gamma carboxyl. Gamma carboxyl. So they are highly acidic. They are highly acidic and both non-essential. Okay, both non-essential. They can easily be synthesized in the body. So after that, what is the next is you just take the amide. Okay, you just take the amide. This can be amidated. No, they can be amidated. So aspartic acid, no, aspartic acid becomes asparagine. Asparagine. Okay, so this is your amide. Your amide. What can we say with your amide? No, your nitrogen. Okay, So nitrogen is highly electronegative. Nitrogen is highly electronegative. The hydrogen here can participate in your hydrogen bonding. So your asparagine and glutamine are what? They are neutral. They don't ionize, but they are polar. Okay, they are polar. Don't worry, Doc. I cannot remember. I'll give you an easier way. Okay, so stick around. You stick around. I'll give you an easier way to remember if they're polar, non-polar, etc. Near the end. Okay, I'm just giving the basis. Okay, I'm just giving the basis. So if you have an amide, no, this is an amide of aspartic acid, and this is amide of your glutamic acid. So they are what? They are polar, but they don't ionize. They are neutral. Okay, please do remember the one-letter name. So you have only four. If you have acidic amino acids, what are these three? What are these three? Histidine, arginine, and lysine has an additional amino group that can accept nitrogen, that can accept a proton, or that can accept a hydrogen ion. So these are your basic, okay? These are your basic amino acids. Basic amino acids, they accept a hydrogen ion, they accept a cation ion, becoming a cation, becoming positively charged. So this is arginine R. What is the R group? What is the R group? Please do remember. Arginine has some special peculiarities. Okay, so I'll tell you now. Arginine is the most basic amino acids. Now, when we're going to discuss your ionic properties of your amino acids, excuse me, arginine will have the R group of arginine, your guanidinium group will have the highest pKa. Okay, in the next live stream, we'll be discussing the titration of your amino acid. So it's your guanidinium. Okay, please do remember there are only seven. Huh? So please remember the additional functional groups of seven amino acids. Okay, only seven. Then the 13 will tell you that they are neutral. Okay, the trifunctional amino acids are either acidic or basic. Lysine, that is K. Okay, that is K. What is the functional group? Okay, let's count. If this is alpha, this is beta, this is gamma, sorry, this is alpha, beta, gamma, delta, epsilon, okay, epsilon. Or the amino group is attached 1, 2, 3, 4, 5, 6, no, to this 6 amino, okay, 6 amino or epsilon amino. That is epsilon amino. That is epsilon amino. So this is the 6th trifunctional amino acid. And histidine, okay, histidine, this can be protonated, okay, this can be protonated, and we know very well that nitrogen can only have 3 bonds, right? So you have a positive electrons, this can become positive. No? What do you call this group? What is the functional group of histidine? Functional group of histidine is, the functional group of histidine is what? Is imidazole. Okay, anyway, it's here. Is imidazole, no? Is imidazole. So we have 19 of the 20. Okay, what is the last? The last is the bunso. Okay, the bunso. By the way, this is to show you, that histidine, no, the pKa is near 6. No, it's near physiologic. No, later on you will learn that histidine is a very important component of your buffer systems because its pKa is near your physiologic. And don't be confused. Doc, the pKa is below 7. Why is it basic? No, because remember the Bronsted definition of a base. Okay, it can accept a hydrogen ion. Okay, it can accept. It's a proton acceptor. Then the last is your proline. No, proline is special. No, because look, what is the R group of proline? The R group of proline is what? No, your R group is joined to your alpha amino. Okay, so you have a pyrrolidone, but you have a pyrrolidine ring. Now I'll check it up. No, but when you have a ring, so this is an amino acid, and that's an amino acid. And you remember in a certain anhydrine reaction, it will give a different color, right? It will give a different color. Okay, so please do remember, huh? Please do remember this. No, there are only seven. It will really help you remember in the exercises later on. Okay, so what is the so all amino acids again? No, 20 amino acids will have at least two functional groups. Okay, so 13 of the 20 amino acids are difunctional amino acids. Difunctional. You have a second amino. You have two functional groups. You have your alpha carboxyl and alpha amino. Okay, so that's 13 of the 20, but you have 7 amino acids that have a third functional group. They have an additional functional group. That's why they are rendered either acidic or basic. If the functional group can donate a hydrogen ion or can donate a cation, they are acidic, right? But if the functional group can accept a hydrogen ion or a cation, they are basic. Again, no, I hope you remember what is the functional group, the third functional group or the R group of aspartic acid. It is your beta, okay, it is your beta carboxyl, okay, beta carboxyl. Beta carboxyl. Your glutamic acid is your G, no, GG, okay, that's your gamma carboxyl. Cysteine, what is the third functional group of cysteine? It contains your SH, right? And hydrogen can be donated. It is your sulfhydryl group. Tyrosine is you have a hydrogen OH, no, you have an OH attached to the benzene ring. You have pi electron delocalization. The electron will more have a party with the electrons, with the pi electron delocalization of the ring. Hydrogen can be donated. So tyrosine is phenyl hydroxyl, and all four are acidic. Okay, all four are acidic. Then arginine, what is the third functional group? Again, remember arginine is the most basic. Okay, so most is the strong is the most is the strongest or the most basic. Okay, or the most strongly basic amino acid. Arginine is your guanidinium group. Okay, lysine is your alpha, beta, gamma, delta, epsilon. That's your epsilon amino, and histidine is your, okay, your imidazole. Okay, your imidazole group. So I think we're now ready. So in order to make it easier, okay, in order to make it easier for you to remember how they are classified structurally and based on their polarity, because their property is what? No, is a function, no, of their structure. No, that's biochemistry, okay? So basic and acidic, no, from the Bronsted definition is these are the amino acids that can ionize. No? If they ionize, what does that mean? They become ions, right? If they become cations, if they become cationic after ionization, what happens? They donated the hydrogen ion. They are acidic, right? They are acidic. So they are, sorry, so they become negatively charged. No, correct myself. Negatively charged, they are anionic because they donated the hydrogen ion that is a proton. However, if if the amino acid will accept a hydrogen ion or a proton, what happens? They become cationic and they are basic. So acidic, so this, what you can see here, the acidic and the basic amino acids are the seven. So the neutral amino acids actually are the 13. No, you don't really have to remember. What you have to remember are the seven. So acidic, you have your aspartic acid. No, that's the strongly as it's the strongest, it's the most acidic amino acid. Okay. Then your glutamic acid. Then you have your two weakly acidic amino acids, your cysteine and your tyrosine. So it is prudent to please remember the functional group. That will help you remember. Okay, that will help you remember. Cysteine, sulfhydryl, it has sulfur. It is very highly electronegative. Okay, so that is acidic, right? And what is basic? Basic. So basic, I'll give you a mnemonic. Okay, so please excuse me. Okay, just for you to remember. So you have your histidine, which is weakly. Now, what is the functional group of histidine? Your imidazole group. You have your arginine, that is the strongest amino acid, that is your guanidinium group, and your lysine. So this is your HAL, no, your basic hull. Okay, so I remember this, no, when I saw the movie Shallow Hull. No, so just forget Jack Black and Gwyneth Paltrow. Just remember basic hull. Okay, so they are your basic amino acids. Okay, basic amino acids. Okay, so just remember these seven. These are your basic and acidic amino acids. So the rest are neutral. But the point of contention here is if it is neutral amino acid, it does not mean that they are non-polar. No, because neutral amino acids can be polar. There are groups can contain um, they contain your highly electronegative atoms like nitrogen and oxygen attached to a hydrogen ion that can participate in hydrogen bonding. Okay, so if you remember in our discussion, what are the neutral amino acids that can participate in hydrogen bonding, or that cannot ionize but are not nonpolar or but are polar. If you remember, you have two amino acids that contain your hydroxyl group. They contain your hydroxyl group, and these are your serine, right? Serine. It contains your hydroxyl group and your threonine. So serine and threonine are hydroxyl containing amino acids. They are polar neutral. They are polar neutral. It means that they don't ionize, but they can participate in hydrogen bonding. Thus, they are polar. Then what else? What else? What do you remember? What do you get from if you amidate? If you amidate aspartic and glutamic acid, no, the additional carboxylate group is amidated. No, it's amidated. So aspartic becoming asparagine and glutamic becoming glutamine. No, your amides. These are polar functional groups. Remember, amides are polar functional groups. So just remember these four: two hydroxylated and two amides are polar neutral amino acids. Then the rest, so these are 7 plus 4, that is 7 plus 4, that's 11. So the 9, tama ba? Tama. The 9, you don't have to remember. Don't remember anymore. Okay? Don't remember anymore. But from here, you can see that these are your aliphatic amino acids, right? Your aliphatic amino acids, including proline, your phenylalanine and tryptophan, they contain benzene rings, no, without any polar substituents, and proline is the bunso, no, it's an amino acid. Okay, but for me, just remember the 7 and remember the two hydroxyl containing amino acids, okay, and the two amide containing amino acids, and they are the polar neutral amino acids. So you're done. Okay, so essential, no, I think we're nearing the end. We're nearing the end. Essential amino acids. These are amino acids that are required in the body. Okay, that they are required in the body because we lack the enzymes. We lack the enzymes to synthesize them. No, they're harder. Okay, they're complicated. For example, branched chain amino acids are very hard to synthesize. Okay, non-essentials, they need not be supplied in the diet because they can be synthesized. Now we're armed with all the enzymes, to synthesize them. Okay, so this is another mnemonic. Huh? So we have I didn't fulfill my promise. I use mnemonics, but you have to. Okay, no. So it's a movie. No, maybe you were not yet born. Anyway, so the mnemonic really for your amino acids is your private, no, private team hall. Okay, private team hall. Okay, so these are phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, lysine. But the problem with mnemonics is you know the mnemonic, you know the letters, but you don't know which they stand for. For the L and L, we don't have any problems because you have only two amino acids that start with the letter L, right? What about T? This is the problem. This is the problem. You have three amino acids that start with the letter T. You have threonine, tyrosine, tryptophan, right? The thing to remember here is that threonine is a branched chain amino acid. Okay, so it's hard. Then tryptophan with the indole, right? It's structurally complex, right? So it has to be taken in. Okay. Or if you want, tyrosine is just a hydroxylated phenylalanine, right? If you remember the structure, so my advice is you look into the structure, you try to remember. You don't really have to memorize the structure and draw it by hand, but you can if you want to, but just look at the special features. Okay, tryptophan is an indole. This is really very complicated. Threonine is branch. Okay, so one of the four branch chain amino acids. Okay, and tyrosine can easily be hydroxylated from phenylalanine. But what I want to emphasize here is there's this not really a controversy, but a qualifier, a caveat. Okay, if we're going to count this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, you have 10, but this is not always. Okay, you don't always absolutely have 10 essential amino acids. No. You don't always have 10 essential amino acids because what? Arginine is only what? Arginine is conditionally essential. Normal adults, normal, healthy, non-sick, okay, non-sick adults, we can synthesize arginine, no, in the urea cycle. However, infants, growing children, recuperating patients, no, from surgery, arginine becomes essential. Okay, so I told you arginine is special. Arginine is special. No? It's the most basic and it's essentially conditionally essential. Okay, so it depends, but a lot of books do agree. A lot of books do agree. Okay, so these are your essential amino acids. Then don't remember the others. Okay, so the amino acids that are not mentioned here are the non-essential amino acids. Okay, any more but the T and this is the problem, the T. The P, private, you don't put P because you might confuse it for proline. Okay, just use F. Then amino acids that can be synthesized in the bodies, these are your non-essential. Okay, why is so that's the rest. You don't really have to remember. Okay, just remember the essential. Again, arginine is here. Why? For normal adults, non-sick adults, okay, your arginine is what? Is non-essential. You can synthesize that. Cysteine, as I've mentioned, cysteine becomes essential in what condition? Cysteine becomes essential if methionine is deficient. Okay, we know that methionine is essential, right? However, no, for starvation, etc., no, for one reason or another, methionine is deficient in the diet, cysteine becomes essential. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10, okay, so 10 from glucose, and you can see what is missing. What is missing? One amino acid is also non-essential because it is synthesized from phenylalanine. Okay, sorry, it is synthesized from an essential amino acid, right? That is your phenylalanine, that is your tyrosine. Okay, so tyrosine. So from glucose and from essential amino acid, your cysteine is just sourcing, no, sourcing the sulfur from your methionine. Okay, so let's do some exercises. Okay, sorry, I overextended my time. Okay, so feel free to answer, and later we can have a Q&A. So I know, no, you Doc, it was too fast, no, but just go back to this video. But for me, the point here is if you're going to remember the mnemonics for the classification, it's really very hard. No? Just look into the structure. Just try to remember the specific, peculiar, okay, uniqueness of the structure, and it will help you. Okay, so question: this amino acid is classified as non-essential in the diet. Okay, with an R group, non-essential. Doc, what I remember are the essential, right? Anyway, we have some clues here. With an R group that is polar and does not ionize. What does that mean? It does not ionize, so it does not involve the seven, right? The seven. It does not involve the seven, diba? The seven, aspartic, glutamic, cysteine, tyrosine, the hull, histidine, arginine, lysine, because they are the ones that ionize, acidic and basic amino acids. So aspartic acid is out, right? So you can wait. So you can scratch this out, right? You can scratch that out. Okay. Polar. What is polar? So what we're looking here now is a neutral amino acid that is polar. Okay, a neutral amino acid that is polar. So what do we remember again? A neutral acid. Ito, histidine again. Histidine is what? No, histidine is imidazole. That's hull. So this is scratch as well. Scratch as well, right? Because if you're going to look at the question, it is intimidating, right? If you remember the mnemonics, it will next impossible because the question is confusing as well, right? But if we're going to to look at it, no, to analyze it step by step, no, it will become easier. Okay, we know very well that serine and threonine are what? They are polar, right? They're polar, right? They are polar and they are neutral, right? They do not ionize, but they are polar. But what is the answer now? The what will since the answer is this, no, non-essential, non-essential. Which is non-essential, serine or threonine? Which is non-essential, serine or threonine? Threonine is branch, right? Threonine is branch It is one of the T in private team hall, right? Private team hall. So the answer here is serine. Okay. So you look into the question, but if you look at the question in the exam, okay, it will really be confusing. Oh, same in the same vein. I hope you can follow. No, this amino acid is classified as essential. So we can now look into the private hemol with an R group that is polar and neutral. So more or less the question is the same or similar, right? So you can do it the other way around. No, you essential. You look at the essential. I know the mnemonic there. The mnemonic there for essential. Is serine included in the essential? No, no, it's not private sim hall. No, you don't have a letter S, so that's not there. Okay, is tyrosine essential? Tyrosine can be what? Can be hydroxylated from phenylalanine, right? So please remember the two T's in private hemol is threonine, a branch chain, and tryptophan, the indole. A highly what? A um, highly complicated structure. So you're left with histidine and threonine, right? Private sim hole, okay? Hall, private sim hall, and this is one of the T's. So we're left with two, okay? So by scratching out, no, by process of elimination, that is polar and neutral, okay? Both are polar, right? Both are polar. Actually, one ionizes, one is not. Eh. So neutral means that it does not ionize, okay? So from the two, which does ionize? It's your hull, right? Histidine is basic. Histidine is basic, so this is scratch out, right? So did you get it? Did you get it? Essential, we already scratch out two. Polar and neutral, we scratch out histidine because histidine is basic. Histidine can ionize, right? So negating this or precluding this. So what is polar, neutral, and essential is threonine. Is threonine, okay? This one, okay. In a globular protein, for example, in myoglobin, you know very well that myoglobin is a protein in the muscle. No, it's an oxygen transporter. Okay, transport carrier protein. Okay, which of the following groups of amino acid is located in the exterior portion of the molecule? Exterior. Again, you know very well that water is life. Okay, universal solvent. But some proteins should they should be in a way soluble, right? And what makes them soluble is because they have your polar groups that are located outside. So all the non-polar, all the hydrophobic amino acids, okay, how many hydrophobic amino acids do we have? How many hydrophobic amino acids? I know that you have seven trifunctional amino acids, right? You have four acidic, three basic. Okay, so they are polar. They will be located outside, right? They will be located outside. Okay. Then you have your neutral polar. Your neutral polar are your hydroxylated amino acids and your amidated amino acids. Okay, the question here is in the exterior portion. So exterior portion, in this case, we're looking for what? We're looking for the hydrophilic amino acids. No, hydrophilic. So the hydrophilic amino acids can either be amino acids that ionize. No, in other words, they can form cations and anions that can participate electrostatic in electrostatic interactions or ionic bond formation. No, ionic bond formation. So what we're going to look here now is what? No, what we're going to look for are choices that will have all hydrophilic amino acids, right? So letter A, methionine, valine, proline. Methionine is strongly hydrophobic. Valine and proline belongs to the first group, no, your aliphatic nonpolar amino acids. So this is scratched out, right? Okay, phenylalanine, tryptophan, methionine in the same way. The methionine is there. The tryptophan and phenylalanine, they are what? They are hydrophobic. No, phenylalanine is one of the most hydrophobic. I mean, sorry. Sorry. Okay, I pressed the mouse. So this is scratch out, right? Arginine is what? Arginine is the most basic. Lysine is a strongly basic amino acid. Aspartic acid is the most acidic. So all of these ionizes. this becomes what? No, this becomes positive. Okay, they have an additional amino group that can accept a proton. This can donate a proton. It becomes negative. So they ionize. They can form ionic bonds, right? They're hydrophilic, so they are what? No, located in the exterior portion. In this case, what? No, the valine isolation and leucine. These are very, very hydrophobic. No, they are what? They are branched chain amino acids. Okay, so the answer here is your amino acids that ionize. Okay, amino acids that ionize. These are your acidic and basic amino acids. Oh, in a globular protein like immunoglobulins, which of the following groups of amino acids is located in the interior portion? Okay, so this is just a counterpart. Interior portion. So in order to protect the amino acids, no, if they're hydrophobic, no, they should be cloistered, or they should be sequestered inside, no, in the interior of the globular protein. So what are we going to look for here? We're going to look for amino acids that are, ayun pala, thank you very much. So okay, no, so in the interior portion, sorry, no, I didn't see the answers. Okay, so for this, you may answer. You may answer. Okay, so in this case, we're looking for amino acids that are hydrophobic. Cysteine, aspartate, glutamic. These are what? This is slightly acidic. Okay, this is. Do you see my laser pointer? Okay, this is slightly acidic. This is strongly acidic, and you were looking for hydrophobic amino acids, so this is scratched out. Okay. Phenylalanine is hydrophobic, right? Tryptophan is hydrophobic. Valine is hydrophobic. If you're not sure, let's look at the others. Okay. Asparagine is what? Asparagine is an amide of aspartic acid. So this is polar. This is polar. So this is hydrophilic. Glutamine is an amide as well. Okay, so it is polar. Although it is neutral, it is polar. Tyrosine is weakly acidic, right? Okay, very good. No, so thank you also, Ezel. Very good, fantastic. No, John Lester. Okay, John Lester, very good. John Beltran, very good. No, so at least I'm very happy that we're hanging on. Oh, histidine, isoleucine, and leucine. Isoleucine and leucine are branch. Doc, pwede yan ah. This can be. But histidine is basic, right? So from here we can see, wait, sorry, that this cannot be, so the answer here is B. Okay, the answer is letter B. So Ezel, okay, very good. Okay, John Lester, very good, and John, very good. No? So you're getting the hang of it. Okay, which of the following statement is true of amino acids? Which of the following statement is true of amino acids? All aromatic amino acids are essential in the diet. What do you think? What do you think? All aromatic amino acids are essential in the diet. What are the aromatic amino acids? Aromatic amino acids are what? Tryptophan, tyrosine, phenylalanine, right? I know that phenylalanine is a very essential amino acid. Same with tryptophan. What about tyrosine? Tyrosine is what? It can easily be what? It can easily be hydroxylated from phenylalanine. Okay, so letter A is, I think, is not. It's not. Okay. The main natural, very good. Okay, so Alvin, right? No, Alvin, right. Tyrosine is not essential. Very good. Among the amino acids, tyrosine, among the aromatic amino acids, tyrosine is not essential. Okay. The main and naturally occurring form of amino acid in mammalian proteins is the D enantiomer. Is the D enantiomer. The main and naturally occurring form of amino acid in the mammalian protein is the D enantiomer. True or false? True or false? It is false. No, D enantiomer is for carbohydrates, right? Um, L enantiomer is for amino acids. So this is wrong, Timon. You have a lot of wrong answers. Amino acids with an amide R group Amino acids with an amide R group are nonpolar but ionizable. Amino R groups with an amide R group are nonpolar but ionizable. It is wrong in both ways, right? Amides, no, same with hydroxylated amino acids like serine and threonine. Amides, you have your asparagine and glutamine. They are neutral, no? They are neutral. They are neutral, so this is wrong. And they are non-ionizable, right? Okay. So they're polar, sorry. They are polar and they are neutral. So this is wrong in both counts. Okay. So you're right. Again, right? No. So John, Lesser, Ezel, and Ezel. Okay, you're right. Letter D. Acidic amino acids exist as anions in physiologic solution. Acidic amino acids exist as anions in physiologic solution. What is a Bronsted acid? A Bronsted acid is a moiety that can donate a hydrogen ion or a proton, right? So it will become what? It will become an ion, right? So in physiologic solution, and we're dealing with the carboxylate. So this is wrong. This is wrong. This is wrong. So our answer is what? No, I think this is correct. Okay, so the answer here is letter D. So wait, what's next is okay. So thank you very much. That ends. So I think I over abuse you. You're welcome. So thank you very much, everyone. Do you have any questions? Do you have any questions? If you're getting some value, because proteins are life. Yes, this is right. Proteins are life. Huh? Proteins are life. They give you muscles. Okay, so you'll not interchange that with carbohydrates. If you have any questions, or if you find value in this video, no, kindly subscribe and maybe like the video so that it can reach more people, and maybe share this as well, not to your classmates. Any questions? Okay. So again, so thank you very much, no, for the 18 million for the 18,000. No, I do appreciate. This is my first live stream in my life. Okay, first live stream in my life. So I will not record videos anymore. Now, if you look into my videos, they're recorded and they're unnatural, right? So welcome Alvin. Okay, welcome Alvin. Maybe you can you can what you can do is you can post a request. Okay, so what do you want me to discuss? I know I have something that will be coming up. No, what will be coming up? I think is the ionic properties of amino acids. It's one of the most confusing. No, in Filipino, masalimuot. Okay, the ionic properties, titration of amino acids. I think that will be next. No, maybe middle of next week. Okay, middle of next week. Do you have any comments? Okay, so thank you, Jacobs. Okay, thank you very much, no, Andrew. Really appreciated. Ezel Benitez. Okay, so thank you very much for answering for those. If you have any requests, no, you can comment on the video. Anyway, this video will be recorded by YouTube. Yana, welcome. Okay, so welcome. I know I went over time, okay, but this is my first live stream, no, so thank you for bearing with me. For the ionic properties of amino acids, no, I'll try to make a lecture that will make it more understandable. Okay, and anyway, we have discussed your Bronsted definition of an acid and a base. But what will really be very confusing are the trifunctional amino acids, again, the seven. The 13 amino acids, the difunctional amino acids, you don't really have any problems. But what will really be confusing is because your third functional group will have a different pKa value. No? So it makes your titration, their ionic properties very complicated. Okay, so with that, do you have any requests? So welcome again, Yana. No, so from the bottom of my heart, thank you very much for staying up this late, and I'll see you soon. Okay, so thank you and bye bye.